And then another thing, those who are unable to log in, please type in your student number so that I can key in your attendance later. Right? Okay, 106. So I'm going to stop sharing the QR attendance. Okay, can you see the slide for today? Chapter
Yes,
10 genomics.
Yes, doctor.
Okay, good. All right. Today is topic 4. Topic 4, which is on genomics. Okay, you have done topic 3 on genetic diseases. So this one is a continuation, a slightly different topic because you imagine once we previously people have already right. The topic for today is genomic. So previously we discussed on mutations on certain gene that cause disorder in human, and the one that we discussed is a single gene mutation that can that can cause disorder. But today we are going into a bigger picture in which we want to learn about genomics, right? So uh, anyone come across what is the meaning of genomics? Something related to a genome. Anybody have any answer to that? What is genomic? Study of genome, okay? And then, okay, then someone mentioned, is it gene modification? Okay, and then the another answer is study of human genome. Um, Actually, it is everything related to genome. Okay, it's not only on in human genome, but any study related to genome of any organism. So that will be study of the genome. So genomics can cover all organisms as long as they have a genome. Okay, so that is genomic. And inside, once you study genomic, you know the gene structure and then the gene function, everything. You will know when you know about genomics. I mean, know in detail because previously even without the information about genomics, people can still study gene. So not to be confused, or you cannot say that it has to be in a specific order, right? Because you have to remember, prior to the study of genomics, people have been studying about genes for quite a long time already. Okay, so genomics, the definition, branch of molecular genetics concerned with the study of genome, specifically identification, sequencing of their constituent genes, and application of this knowledge for many in many area, in medicine, pharmacy, in all scientific area, as long as it involve organism, then you can use the genomic information. Uh, Genomics can also be described as study of all the nucleotide sequence in organism. So with the nucleotide sequence, you can learn about the gene. We call it structural gene, and then a regulatory sequence, genes that can allow to express certain protein, to control certain protein, certain enzyme. So those are regulatory sequence. Um, and then in addition to that, studies have shown that our DNA, especially in eukaryotic organism, not all sequence or not all nucleotide sequence is a coding sequence, which means that there are area that is what you can describe as a non-coding DNA sequence or segments in the chromosome. Okay, so anything you just imagine genomics, if the organism has the DNA, then in all chromosomes, and we want to study that, so we are studying the genomic. So we are doing the genomic study. Okay, so from Nature magazine, actually is a journal of scientific eminent, or um, the the highest achievement that one can go in a scientific world if you want to publish your result or your scientific discovery. So you publish in Nature, right? So this is the highest possible level usually people go for if they want to publish. So it can be in Nature Genetics, Nature Plants, and Nature, but the highest level of all is Nature. So that is a journal for those publishing scientific result. So the Nature described genomic as the study of full genetic complement of the organism. So I'm quite sure by now you have already understand in terms of when the organism has the DNA sequence inside it. So it is called the full complementation or full um, collection of the genes inside it. So you call that the genome. And then those how do you study the genome? In many ways. So it employ a recombinant DNA sequencing method, and then you use the bioinformatics to do the sequencing, and then you use the bioinformatics to assemble and analyze the structure and function of the sequence of DNA in any organism. Okay, so that is genomic. So again, the idea of genomic is that it is related to genome, and genome means that full complementation, full content, full constituent of the DNA in any given organism. Everything, all the DNA sequence inside there. Okay, and genomics is uh, uh, part of the omics study. And then in genomics, there are many uh, further derivation of the omics. Okay, for example, in computational genomics, you are studying the DNA sequence of organism, but more of using algorithm programming. And then Metagenomic can also relate to metabolism. Epigenomics can relate to differences between the same organism but have different expression level, for example. So that one is epigenomic. Nitrogenomic, actually, this one is not nitrogenomic. It's more of a like nutraceutical, so genomics are something related to the nutrients, the nutrition. Okay, and then whole genome sequencing is usually when you sequence everything with the DNA constituent of that organism. So many of these are related to genomics. And then from the genomics, you can, with the genome, you can study the gene structure. And then from the gene structure, you can translate into protein. So you go into proteomics. But the actual study of proteomics is slightly different, where you study the protein collection. So actually, proteomics is not in genomics, but actually it is its own study compared to the others. Like all of them got genomics. Suffixes at the back, okay, but then for proteomic it is a totally different study, okay. And then genomics of domestication, for example, people has studied for example genomes of the dogs, uh, genome of certain plants, okay. Domestication means that usually it was wild organism, okay, the wheat, for example, the dogs, for example, and then somehow throughout the evolution, changes in the gene sequence allow those species to be improved, for example. So that one is a genomic of domestication. And then immunomics related to the genes that has to do with our antibodies, in immunological characteristic of the individuals. So those are in immunomics. Okay, DNA sequencing techniques. So the advancement of the DNA sequencing techniques change the experimental biology, and automation has enhanced the rate of change. You just imagine last time when the simplest imagination you can say is that before this during certain time when you study one gene only, you study one gene, you remember a gene has to consist of for example it start with the start codon, right? ATG, and then it ends with uh, uh, what is the stop codon? GTA, for example, GAT, for example. So, and then all these are open reading frame. We will come across open reading frame. So you study one gene, you can get a PhD. Okay. And then during my time in the late 90s, like my own example, I study three genes, for example, three genes related to starch biosynthesis, for example, three genes plus proteins characterization from this gene. I get PhD. But nowadays, with the advancement of DNA sequencing technique, you study the entire genome of the organism. For example, you uh, then you get a PhD, for example. So these are, you can see in terms of advancement, because last time when it was very difficult to do sequencing of a gene, then it is very difficult, the process, and then the knowledge is quite limited at that time. So that is in the 80s, 70s, for example. And then in the 90s, then automation has started, sequencing become quite fast. So in my case, for example, three uh, is more or less sufficient, 3, 5 intense, for example. But then nowadays, usually people study the entire genome in certain area lah. Of course, I'm talking about if you are in the genomic or molecular biology area, for example. And of course, this one, don't be alarmed by it. This one is a general situation because there are also other study whereby you have to go into very specific gene and enzymes and then study their biochemistry, everything. So that will be different. This one is just an idea for you to see how advanced it has been going for the last 30, 40 years in terms of knowledge and technique related to sequencing and the genome. Okay, so genomics is the development of application of techniques for mapping chromosomes, sequencing the genome, and then analysis of the genome using algorithm or computational method. Okay, so that is how the advancement of genomes has been going on for the last, I think, 30 years. All right. So mapping, and then can you imagine what is mapping? What is sequencing? Mapping, you just imagine like on the chromosome. Okay, let's say this one is a long chromosome. Okay, very long chromosome and then this is the centromere, for example, over here. And then with the mapping of the chromosome, you have general idea. Oh, the the gene related to the eye color is over here. The gene related to the length of the limb is located over here. But then you don't know what is the actual sequence, for example, or sometimes you just know just a little bit of the sequence. So that one is mapping. And then and that's with the advancement of technology, advancement of the technique the scientist has obtained, then you can start to get what is the ATGC sequence of the gene, for example, of this gene, and then certain area. So mapping related to the general area on the chromosome, sequencing more detail, and then once more advanced, go into the technique of sequencing, then you can sequence the entire chromosome. Okay, then once you have this sequence of ATGC for entire chromosome, you become not stuck, but then you just want to analyze everything. It is very, very difficult because so many nucleotides. Then you develop a computer algorithm and so write a programming to analyze the sequence. So then based on the program, you determine that, oh, on this chromosome, based on the analysis, actually there are several genes. Okay, on throughout the chromosome, actually there are 25 genes, for example. Okay, and then you want to restudy each gene again, for example. So these are the process of how study related to genomics. Okay. Okay, and then again, genomics have the subfields or the subcategory in terms of the study of the genome. Okay, one is structural genomics. So structural genomics, you don't get confused with the structure of a protein. Remember, in our previous lecture, we are talking about structure of the protein. You know, like the hemoglobin, genes produce a structural protein. So in that case, that structure protein is physical, although very small in nanometer. It's a physical block or physical existence uh, of uh, of a molecule. So that one is protein structure. But when we are talking about structural genomics, then it is actually an abstract in which you know, like I mentioned in the previous slide, on the chromosome, okay, and then on you have genes, okay, and then on the genes you have, for example, like start codon, start codon and stop codon, and then the open reading frame, and then you study that, oh, on the gene, this one is a regulatory gene, this one is a gene that produces an enzyme. So you know that on the chromosome, okay, this is located over here, this is on the P arm, this one is on the Q arm. So those are what is meant by structural genomic, in which it is related to genetic and physical mapping and sequencing of the chromosome. So when you do a sequencing of this chromosome, this gene, this area of the gene, so that is what is meant by structural genomics. So it's not like it's a molecule, okay? And then functional genomics is another one. Functional genomics, one is okay, one is structural, the other one is functional genomic, comprehensive analysis of gene function and non-gene sequence in the entire genome. So this one, as the name implies, it is related to what is the role of that particular sequence on the chromosome. Okay, for example, this area, what is the role? Does it have a sequence, a gene that control production of this gene, for example? So analysis of the gene function. So what is the function of this gene? What is the function of this gene, and so on. And then is there, when you study this sequence, for example, although this one is not a gene, does it play a role? Okay, so that is the functional genomic. For example, another example is like a repetitive sequence on certain area that can be used for DNA fingerprinting, for example. I have 3 repetitive sequence of, for example, GGG, CCT, GA. So I have 3. And then if we do analysis, oh, another person have 4, another person have 5, another person have 3 similar like me. So those are what is meant by functional genomics. And then the other subfield is comparative genomic. This one, as the name is saying, you compare. Comparison of entire genomic across the species, looking at function and also evolutionary relationship. Okay, for example, like you probably still recall a picture of a chromosome, okay, and then got very dark, and then a light area, and then small dark area, light area, dark area, short dark, and then long dark area. And then this one is in human, and then in chimpanzee, same pattern like this, but on different chromosome, for example. So comparative, you compare the sequence between or the pattern of chromosome or the pattern of the genome sequence between different species, or sometimes within the same species, you see whether there are differences between plant that produce a very tall plant, and then the same species but not growing very fast, so you compare. So that one is also a comparative genomic. So when you compare the genome, so that will be comparative genomic. Okay, but our lecture for this course is focusing on structural genetics, and we go towards genome sequencing, how genome sequencing is done. Okay, so it is more on structural genomic for this course. Okay, but bear in mind, based on the structural genomics, you got the information of the sequence, the entire sequence of the genome, then you can study the other thing as well. Okay. Any question so far? Chat box? None. Okay. So we continue. Okay, structural genomic, characterizing, locating the entire set of genes in the genome to characterize the structure of the genome. So again, as I mentioned just now, the structural genomics related to, you want to know the sequence of the entire uh, chromosome collection in the organism, and then based on that, you analyze into more detail, so that those study is a structural genomic. And knowledge and structure of an individual genome can be useful in manipulating genes and DNA segments in that particular species. So this one, what I can say is that we are specifically not referring to human genome, because for human genome, it is entirely what you call that unethical to do any manipulation of our gene, for example, right? So you don't do manipulating, but then in plants, for example, in microbes, for example, then we want to study how to, let's say, have the bacteria to produce more hydrogen, for example, because we want to go towards green technology, car using hydrogen as fuel, hydrogen fuel, for example. Then you want to use biological method to produce hydrogen. Then you manipulate the certain genes of the microbe to produce. So that is manipulating genes and DNA of a particular species, for example, right? Okay. At the moment, if you have any question, please you can always put it in the chat box. Once in a while, I'll have a look and see if there's any question related to the topic that I'm talking about today. All right, when a number of genomes have been characterized at the structural level, the hope is that through comparative genomics, it will become possible to deduce the general rules that govern the overall structural organization of all organisms. So again, as I mentioned just now, if you go back to the previous two slides, for example, from the structural genomic means that you know the characteristic, the sequence, and then based on the sequence, you know this one is a start codon, stop codon, and then what is the function of the genes, So those you can study further, and then you compare comparative genomics, for example. So it will become possible then to find out what are the general rules that govern all the structural organization of the genome. Okay, for example, another example that I can give you so that you can visualize. Okay, for example, in human, you have a certain gene that you don't know, but you know the sequence already. Okay, and then in yeast, you have the chromosome of the yeast, and then this sequence you know, and then you know the function of this gene. So then you can do a comparative genomic. So you can see in human, because the sequence is very similar to you have in yeast, then the sequence that initially we don't know about what is the function of this gene, because they are, for example, like a 99% similarity with the yeast one. Then we can say that this unknown sequence in human is actually doing similar function as the gene that we have already described in the yeast. Okay, so that is the comparative genomics. So you can deduce the function of the genes that was previously unknown. Okay, so that is how scientists do the study, because most of the time we cannot uh, directly study sequence in human if we are doing human genome sequencing. But then we can study the genome in fish, for example, in rats, mouse, in other organism, and then when we compare certain genes, they are very similar to human. Then you can deduce the function of the human gene actually is same as the function that was shown in the mouse, the rats, the bacteria, like that. Just similar like I am showing you just now. So structural genomics proceed through increasing level of analytic resolution. Okay, analytic resolution starting with the assignment of genes and markers, okay, into the individual chromosomes and then mapping these gene and markers within a chromosome. And finally, preparation of physical map culminate, culminating in the sequencing. So this one actually in one sentence from here to here, encompassing several resolution that taken more than 10 years to achieve from uh, genes and markers towards sequencing over here. Okay, so it takes many years. So what is meant by this is that previously we have chromosome collection in Drosophila, for example, 4 copies, right? Chromosome 1, 2, 3, and 4, diploidic Drosophila. Actually, the number 4 is actually XX, for example. So initially, we know only the general idea. Okay, if we have a mutant Drosophila, um, that produce white eye color, for example. Then we analyze the chromosome. We found out that actually the difference between white eye Drosophila, and red eye Drosophila is the difference at this area of the chromosome. Okay, so you know that the uh, the gene that is affecting the eye color of Drosophila is in chromosome 1. Okay, so that will be, and then you know a little bit, so that will be a marker on an individual chromosome. And then later on, you know very specific sequence a little bit. So that will be slightly advancement, and then you know the entire sequence. So this is what is meant by resolution. So resolution means that initially on the chromosome, you just know a general idea. Okay, and then when the technique advance a little bit, you have a certain sequence, and then that sequence is related to this area. You call it a marker, for example. So that will be increasing a resolution slightly, and eventually you manage to identify the sequence of this area. Then you have sequencing. And then after that, all sequence throughout the chromosome of this chromosome, you know. So this one is meaning to say the increasing levels of resolution related to the genetic information. Okay, so it's like last time you use you don't have money, you use Samsung A2, right? And then uh, you did well in your SPM, you go into matriculation, Samsung A32, okay? And then when you go into second year, your parents give you um, a present, S21, for example. So you notice that the resolution of the pixel is increasing, meaning in more detail, isn't it? So similar like this one, last time you know just about existence of the gene, but you don't know the information yet on that chromosome, and then you know the entire sequence, and then after several years, you know the entire chromosome sequence. So increasing resolution. Okay, so that is how uh, you imagine. Okay, that's a huge upgrade. Of course, it is a huge upgrade, isn't it? S21, the highest end of the Samsung, isn't it? I'm quite sure some of you already have it. Okay, even though not all of you, right? But then I think it's quite easy for you to visualize, isn't it? Increasing resolution. Yeah. So in sequence, you just know initially on that chromosome, but you don't know more info. But then after sometimes, then you know the detail of it. So increasing resolution. So so coming back to the lowest resolution just now, so genetic and physical maps are made first to provide markers for sequencing. So you know before, so this one is more or less like um, chronological event in terms of doing the genome sequencing of an organism. Okay, so Genetic and physical maps are made first to provide markers for sequencing, and then examples illustrate the logic of this approach in the human genome project. Okay, so what I can tell you here, what is meant by this is that um, before, okay, like this one is a chromosome. Okay, uh, when people has been studying about characteristic of the gene and then study the difference between normal and mutant, they know that okay, if the changes are happening over here, you know, this one related to eye color, for example, and then later on you know a little bit of the sequence over here. So the sequence over here, you know, okay, and then some other study known the sequence over here. So this will be another sequence that unknown. This one is another sequence that unknown. So when this happen, although not uh, entirely known yet, the sequencing in terms of the ATGNC sequence. So this one can be used as a marker. This one can be used as a marker. This one can be used as a marker, meaning to say that even if I cut this into smaller pieces, and then when we check with this sequence, you know that this actually comes earlier than this. This will come will be in the middle compared to this one on the other end. So you know the marker so that if I do analysis of this, and then if this one is in separate pieces, for example, until here only, and this one from here until here only, and this one from here until here, you know that which fragment this one can be arranged, how they can be arranged. Okay, so this one is how it is done. Okay, so that is a genetic mapping of the genome. So mapping is again, as I mentioned earlier, you kind of have an idea, locate where they are, you know. So that is the mapping of the genome. So genetic maps are constructed for each chromosome using genetic crosses and pedigree analysis. Okay, any detectable allele can mark a locus on the chromosome, and crossing over indicates distance between the marker gene. So for example, you want to do genome sequencing of yeast, for example. All right. So, or in human, for example, you cannot do crosses. Right? If you still recall, in human we don't do experiment, right? So how we want to know? Then we do pedigree analysis. Pedigree analysis is that you do an investigation by surveying, and then based on that survey you can analyze you take the blood and then check the chromosome and know that okay, this is the area that is changes between the albinism and the normal one. So that is pedigree analysis. Okay, and so you can do pedigree analysis in human, or sometimes in yeast, or in bacteria, or in dogs or cats. You know, you can do experimental crosses, and then based on that, you will be able to determine a general idea where the marker is located. So any detectable allele, for example, if you cross between the normal and mutant, then you can see if the progeny is um, is abnormal, then you know that, oh, this one inherited from the parent that is abnormal. Then you can see where is it on the chromosome. Okay, so that is detectable allele which mark the locus. Remember what is locus? The location. So you know that, okay, if this is a sequence, we know that this is referring to a certain area on the chromosome. Okay, so that is how you do the study. So based on that, if let's say a hypothetical organism has, let's say, 6 chromosome, okay, 1, 2, 3, 4, 5, 6, right? Chromosome number 1 until chromosome number 6, we are diploid. So actually, we are talking about chromosome 1, chromosome 2. So based on this, you know that marker A is located A1 located over here, A2 over here, A3, and then B1, B2, B3. So all those will be uh, um, the location of the markers on each of the chromosome based on your earlier study through genetic crosses or pedigree analysis. Right? So we know already. So if A1 located on chromosome 1, A6 located on chromosome 1, uh, marker E5, this one is located on chromosome number 5, for example. And then marker E1 also located on chromosome number 5. So if we are given a marker, you actually know the information about the marker. You can also put where the marker is located. Okay, so let's say I take chromosome 1 later on, and then I enlarge it. So A1 is over here, A2 is over here, A3 is over here, A4, A5, A6. Okay, and then I cut them into smaller pieces. Okay, I cut into smaller pieces, so they become segment. Okay, 7 segment, for example. Okay, six, six lah kan. So even if I put them into a tube, okay, later on I will be able to arrange in order where they appear segmentally, because I know when I do, okay, for example, I put in a tube, so all of them in one tube, 6 fragment here in one tube. So how do I know if this one is located here or located here? So what I can do is that I can find the marker over here, A1 or A2, A whatever. So based on this, then later if I do sequencing, I will be able to tell that the sequence that I have over here actually belongs to this segment, because I have the A3s actually over here. Okay, and then when I do sequencing of this one, and then based on the sequencing, I got the sequence information, but then which fragment this one belongs to, and then I look at the marker. Oh, this one is a A2. So then I know the sequence that I have over here actually belongs to this fragment. So eventually I will be able to arrange everything back together. Then I got the entire information of the sequence of the entire chromosome 1. Okay, so that is how it is done. Okay. Any question so far? Anyone?
No, doctor.
No, yeah?
Dekat
Uh, no
inside
question, yeah?
inside the chat box.
May, saya ada kelas, I call you. Hang on, yeah? Some interruption. Okay, so genetic maps are constructed using the chromosome. Based on the chromosome, based on the marker, you'll be able to determine the exact sequence. Then you will be able to tell what is the sequence. So that is the concept of what is being done in the Human Genome Project, the HGP Human Genome Project. So what happened is that a sequence tag site is a unique genomic DNA sequence used as a genetic marker. So what I can tell you is that before the human genome has started around 1995, 1996, what happened before that? You have to recall in chronologically. In 80s, 90s, people have already been studying what gene is related to what disease. Okay, so they know based on the information that we discussed last week, right? Certain gene cause this alkaptonuria or whatever. So because of that, then they know, oh, this one, cystic fibrosis located on chromosome 7, alkaptonuria located on chromosome 2, for example. So they know certain information. So those information can be one of the marker. But then in some area, we don't have the full information, but then we have a small, shorter sequence information. Similar like the concept I drawn just now, and it is called sequence text site or STS, sequence text site. So those is also a marker. Okay, and then and another name is what you call short tandem repeat, because this one short tandem repeat actually is not a gene. Okay, um, so whatever sequence information that is unique to certain locus or position on the chromosome, then it can be a marker. So in this case, the short tandem repeat, for example, like AGG, CTAA, for example, and then you know that in one area of chromosome number 2, for example, this area is notoriously known for this sequence repetitively. So you can see that they are repeated, let's say 20 times, for example, of this one over here, 20 times. So this, and then you cannot find similar repetitive sequence like this in another chromosome, for example. So that will be used as a marker for this one. So short tandem repeat will be a marker for, let's say this one is chromosome 3. This one will be a marker. Okay. And then you can have another information for marker at this area, another information marker at this area, right? So this will be another way of having the sequence information as the marker. So the marker basically can be a sequence of a full-length gene, or if it is a non-gene, it can be a repetitive sequence, like you call it short tandem repeats, or sometimes another sequence that is unique, you call it sequence text site. Okay. So polymorphic short tandem repeats are the best DNA markers for generating genetic maps of sequence text site. So more or less, STR is the subset of sequence text site, right? So polymorphic STR means that the different people or different individual might have slightly different number of repetition. Okay, for example, this person have 20, same chromosome 3, but for individual, different individual, it might have, let's say, 15 only. Okay, so you know that this one is a very unique site, so it can be a DNA marker. So this one is also what you call as the STR that is subset of sequence text site. Okay, and then so every sequence question. So every sequence has its own significant segment. Actually, the other way around. So every segment has its own significant sequence. Okay, so that is the one of the question asked. So every, for example, like this one will be one of the marker, the marker, the marker. So every, this one is considered as one is a segment. This one is a segment. This one is a segment. So each segment have its own significantly different sequence. Okay, each segment has different sequence inside it, so that you know if the sequence is like this, it belongs to here. If the sequence is like that, it belongs to here. Okay, so that is how it is done. So when you have that, you have physical mapping of a genome. So genetic maps generated for some species are sufficient to begin sequencing. But in humans, even the detailed genetic map described above lacks the required resolution. Therefore, a physical map derived directly from genomic DNA rather than analysis of recombinants have been generated. So, so this one just mentioned, okay, in E. coli is quite simple because I think it's about 3,000 base pair. So you can have slightly information here, here, and here, 3 or 4 different places. Straight away, you can sequence the entire uh, chromosomal DNA of an E. coli to get the genome sequence of the E. coli. So you have the study of the E. coli genome. But for human, it is rather difficult because the human genome are very big. The genome size of human is very big. So even if you have, because you have like 24 chromosomes, right? 24 chromosomes of in human genome. Even if you have marker, it's not enough. Okay, it's not enough because 24 chromosome is very quite a lot. So you need to separate it individually. So you have a physical map, individual chromosome. So chromosome 1, take you put in tube 1, chromosome 2 into tube 2, tube 3, tube 4 for chromosome. So you have 24 different tubes. So and in each tube, chromosome 1 is not only one copy, but you can have 1,000 copies, but all of them are chromosome 1. Okay, so now you have 24 physical maps of human chromosome. So 24 physical maps for autosomes plus X and Y. Okay, so human chromosomes, total collection is 24 chromosomes. Okay, chromosome 1 until 23, 22 plus X plus Y. So 22, 23, 24. Okay, so you have chromosome. So in each chromosome, then you can start to develop its own marker. Okay, so that at least when you cut them, then you know, similar like the concept I explained just now. All right. So in humans, there are 24 physical maps, and types of physical map are presented in order of increasing resolution. So resolution, again, as I mentioned to you, in terms of the information uh, uh, of the fragment until the resolution to the sequencing in terms of ATGNC. So for each of these map, how do we create the marker over here? Then we can do like this. Okay, for example, cytogenetic maps of chromosomal bending pattern. So you know previously, like in the chromosome, you can have dark and then light and then dark again. So just like a barcode. So that one is very low resolution because you can only see the pattern. Okay, and then you can also do slightly better uh, fluorescent in situ hybridization map, whereby, for example, you have sequence information from yeast, and those yeast chromosome, probably you know the sequence on it, and then you take this part, okay, this part only you cut, and then this one, see where they bind on the chromosome. Or so if they bind over here, then you know, oh, chromosome, this segment on the yeast, when you bind to human chromosome, it binds to chromosome number 5 at the, let's say, the Q arm, for example, this area. So then you know, because you know this, then you imply the human chromosome also have similar sequence like this. So this area now known as a marker. So marker, but you see the information is slightly better than the earlier one. So increasing resolution. So you can do this in many uh, chromosomes. So you can have physical maps at the level of segment based on in situ hybridization. And then another way to do it by restriction mapping, in which you can treat some of you can treat the chromosome. They produce certain fragments so that you know this chromosome cut only in this restriction enzyme cut only in chromosome number 2 or chromosome number 20, for example. So you have information increasing resolution. Okay, so restriction enzymes that cut are rarely used to either to a large recognition sequence or due to scarcity of the recognition sequence in the DNA study. So I think this one is a bit, I should have uh, make some correction. Restriction enzymes that cut rarely are used. Rarely, that cut rarely, what is meant by this? You remember the restriction enzyme that, for example, like AT, TA, for example. Oh, sorry, AT. No, cannot use. GAA, TTC. Okay, so this one is recognition sequence, right? So if like this, uh, it is it is able to cut at not that many position. Okay, for example, if you have a 6 cutter on a very long chromosome, it only cut at 3 different places. Okay, but if you have another sequence, AATT, which is another enzyme, it only cut at 4 sequence. The probability of this sequence AATT is higher, so it can cut at many places more. Okay, so the more number of letters, meaning to say that less frequent it will be found on the chromosome. So that is what is meant by scarcity of the recognition sequence, as the recognition sequence size is getting bigger. So for map, even rarely cutting restriction enzyme is very complex, and so far has been obtained only for the smallest human chromosome, chromosome 21 mapped with not 21. So not 21 is a sequence that is quite long, so it can only cut on the chromosome 21 of human chromosome. Okay, so that is a restriction map. So you can see over here, this one is just a general location on the chromosome. This one is slightly specific. You know strike information, and with restriction map, you know that okay, on chromosome number 21, there is only one site that is cut with not one sequence. Okay, so you know that this is the mapping. So if you later do the sequencing, find sequence that is related to not one, or it is only position on chromosome 21. Okay, and then radiation hybrid maps uses can also be done. Okay, in which the radiation hybrid maps, you break the DNA uh, of another organism that is known using X-ray, and then you can use that and hybridize to humans, similar like the fluorescent in situ hybridization, and based on that, you are able to determine the marker of that one. So you can see the increasing resolution from A until D. Okay, and then a slightly bigger resolution, or higher, not bigger, yeah, slightly higher resolution uh, is when you know the sequence better. Okay, so once you have the information, then you can do a contig maps. Contig maps or a contiguous maps. Okay, contiguous maps is a partial restriction digestion that produce a large set of overlapping DNA which are cloned into YAG vector cut with a compatible restriction enzyme. Shearing may also be used to make high molecular weight DNA that is blunt end cloned into a YAG vector. Okay, um, like this. Okay, so now you know initially. Okay, let's say if I do a physical map. Okay, physical map, which means that in each tube I got chromosome 1 until chromosome 22 X and Y. So I take only chromosome 1, and then now I want to do sequencing of chromosome 1. But chromosome 1 is very long. Chromosome 1 is very long. Um, uh, millions of sequence. If you arrange ATGC, see all the sequence, and from one end to another end of the human chromosome 1, it got millions of sequence. So it is very long. The machine cannot do the sequencing. So I have to cut into smaller pieces. Cut them into smaller pieces. Okay, cut them into smaller pieces like this. So how do I cut? What do I cut DNA with? Someone? What? How do I cut the DNA? With what?
Restriction enzyme.
Restriction enzyme. Okay, restriction enzyme. So,
So enzyme.
yeah, correct. Good. Okay, everyone. Hopefully you have an idea how do we cut the DNA using restriction enzyme. Okay. Now, with restriction enzyme, uh, is that if I have enough DNA and enough enzyme, what happen is that if let's say, forget about this chromosome 1 for a short while. If I have this one, the fragment of DNA, okay, and it has EcoR1 restriction site at 3 position over here. Okay, this one is EcoR1. Okay, so what happened if I have 1 microgram of enzyme and then sorry, 1 microgram of this fragment of DNA. Okay, 1 microgram means that it is not only one DNA strand, isn't it? So it will be thousands of DNA strand, but all of them are the same strand. Or okay, so thousands of copies. All of them are like this. So these are in one tube. Uh, and these thousands of copies are, when I measure them, they are 1 microgram. So if I have 1 microgram of DNA of this one, and then I have 1 microliter of EcoR1 enzyme, okay, and then I have 1 hour. If I mix all of them and I put at 37 degrees, all DNA, the 1000 DNA will be cut at this, so I can have uh, fragments like this. Okay, fragments like this. Right. So fragment like this, 4 fragments each DNA. So if I have 1000 of this, so I have 4000, right? 4000 DNA, correct? So that is when we have a situation whereby the enzyme can cut all the available restriction site on the DNA. It cut all of them. Then you call a full digestion. A full digestion. Okay. And then in another situation, I can have a situation whereby I don't have 1 hour. I have only 15 minutes. 15 minutes only. So if I put I have 1,000 copies or 1 microgram of DNA, 1 nanogram or microgram of DNA, 1,000 copies. I have 1 microliter, but I don't have 1 hour. I have only 15 minutes. So I put them inside here at 37, but for 15 minutes only. What happened is that on this DNA, the enzyme might, because the enzyme has only very limited time, it cut only at this position. So, but this one, the enzyme not yet cut because not enough enzyme, so it will go to the other DNA first. So it will cut here. This one will produce 1 segment. And then another one over here, right? Because this one is not cut over here. And then for this one, the enzyme might cut over here only, but not here, not here. So I have a fragment this size, isn't it? So when this happen in 15 minutes, not all restriction sites are cut. Only part of all these are cut. Part of the tree, half or only 1/3 of the tree is cut. Okay, like this one cut over here for the other fragment. It is cut over here. Probably in another DNA, it will cut at this position only. So in this case, the enzyme partially cut the DNA. So that is what is meant by a partial restriction. Partial restriction digestion. So just now it was full. full, isn't it? I said full length, full digestion. Another one is partial. Partial because it cannot cut at all positions. So that will be partial restriction digestion. So what happened when I have a partial restriction digestion like this one, I have a fragment this length. Okay, okay, let's say if I can move this one up, follow or not? Yes, it follows. So what happened is that, okay, this one will be this length, isn't it? This one will be this length. And then for this one, it cut over here only, isn't it? So it will be till here, and then another fragment from the same one will be until here only. And then the other one that I drawn over here, it will cut from here, it will here until here, and then another one will be until here, right? So the third one. So you can see they are all cut by eco R1, but they are cut at different position. So they are now overlapping. You see, they are overlapping. So overlapping is important when you want to do the genome sequencing, which I will explain in here. Okay, so what happened here is that when you have a human chromosome like this one we are looking now, oh, so messy. Okay, you get that, yeah? In terms of restriction, partial digestion and complete digestion, full digestion or complete. Full digestion. Sometimes it is called a complete digestion. Sometimes full, sometimes complete digestion. So when you have a partial restriction digestion, it produces a set of large overlapping DNA which are then cloned into YAG vector. So what happened is that you remember the fragment that I drawn just now, okay? Overlapping, okay? So this fragment I can clone into a vector that is called a YAG vector. So this one will be one clone. This one from here until here, clone. This one is another clone. This one another clone. This one another clone. This one another clone, for example. Okay, so I have an entire genome or an entire single chromosome may be represented in a YAG clone library. So a collection of clone number one, clone two, YAG clone number two, three. Oh no, 1, 2, 3. This one 4, this one 5, this one 6. So entire 6 YAG clones are now called a YAG clone library. This one. Okay. Okay. I pause for a short while. If partial digestion is the eco R1 not fully utilized, at the moment, we are all utilizing this. This is they become different fragment, right? So each fragment they are cloned into different clone. So this one will be uh, a library. So this one will be a collection of a YAG library for chromosome 1. Okay, so all of them will be used. Okay, so YAG clones uh, are then assembled into a map either by matching with a fish generated fluorescent in situ hybridization, a generated chromosome map, or by DNA fingerprinting and assessed by assemble. Okay, now I have this. I have this information, right? So let's say you recall the marker just now. So let's say on this chromosome 1, I have the marker position for this one. I have a marker position for this one. So this one is A1. This one A2. So all chromosomes will have A1 and A2. This one A1 is over here, A2 over here. Uh, A2 over here. No, actually A2 over here. Okay. So even if I take them away as an individual, okay, as an individual contact map, so now I do a sequencing of this. After I obtain the sequence, I will be able to rearrange back. I know because of the marker is over here, I know that this belong to this position. When I do a sequencing of this later on, I will be able to tell that, oh, this one belong to the left because it has the marker over here. So that is why it is important. The marker will determine the relative position uh, that is determining where it should be. Okay, so let's say I somehow when I do the sequencing I forgot to sequence this, but then I sequence this. How do I know that this one is located over here? So what happened is that when I sequence this, it has the information similar. It has the similar sequence to this, right? So this one is what is called overlapping. So these are overlapping at here. So I have a position. I will be able to determine. So this one belong here. So you just imagine, let's say this chromosome is longer. Okay, this longer. Then I will be able to tell that, okay, let's say later on this fragment, I do a sequencing and then this area overlap with this one. Okay, so I know that this segment comes after this over here. So that is what is meant by overlapping. Okay, so you can see here, a complete library should yield a complete contact map indicates an order in which the clone fragment occur in a chromosome. Like this. Okay, this one example of chromosome 7. So on the chromosome 7, previous study has determined that this one is a marker. Okay, so all these boxes, these are all marker. Okay, these are all marker. Okay, these are all marker. And then I take this chromosome. It is not only one copy, right? In the tube, there's thousands of the same chromosome in the same tube. And I do partial digestion. And when I do partial digestion, I will get a mixture of fragment size. Okay, this one is one of the fragment until here. And then this one until here. And then a mixture of fragment. And then some of the fragment are like this. So these are the collection of fragment that actually is a yank clone. So we do a sequencing of this. Okay, so when we do sequencing, we obtain sequence information, isn't it? So when we obtain sequence information, you can see that we know we know that this belong to the left hand side of the fragment because of the presence of the marker or confirmation of the marker over here. So you know that this belong to the left hand side. Okay, and then I do another sequencing. Okay, and then when I do another sequencing of this fragment, for example, so when I do sequencing, one is that I will be able to tell that this belong to somewhere over here because of the presence of the marker. Okay, over here, which is similar like this. And another one is that it has an overlapping region. Okay, it has an overlapping region. Uh, you can see the overlapping. Yeah, wait. You can see the overlapping region over here. So this area, okay, overlap with this area. So you know that in addition to the marker confirming the position, the sequence over here, overlapping over here, they should be the same. Okay, and then I do another sequencing of this clone. Okay, and then I do another sequencing of this clone. Okay, so you can see based on this, I will be able to tell this fragment belong here because the overlapping region is here. Okay, and then based on this sequence information, I will be able to tell that it is located to the right hand side of the chromosome because one is the position of the marker, similar like this. Another one is the overlapping over here. So based on this, you can see that all overlapping function is to confirm the sequencing so that they are in the exact position. So you can know that they belong in this arrangement because one is the marker, another one is the overlapping. Okay, so that is why complete library should yield a complete contiguous contig map that indicates the order in which the clone fragment occur in the chromosome. So you can see over here, even when the chromosomes are cut into smaller pieces and each pieces are individually sequenced, we are able to rearrange their relative position based on the information of the marker and also the overlapping that we have. Okay, so that is how you do it. Okay, so it's similar like the concept you have uh, one strand over here. Okay, okay, and then you have red color over here, and then you have a blue color over here. Okay, and then probably if I have a green color over here. Okay, and then I cut them into smaller pieces. Okay, I cut them into smaller pieces so you can have one strand over here. Okay, and then another pieces I cut from here until here, or sometimes I got three of the similar pieces I cut here until here. So I will be able, if this one got a blue color, okay, if I sorry, no. If I have a green color, without bothering to find out where the blue is here, because they are on the strand, I know that this can be positioned because they are green color, it belongs to the end part, and then this one, because it has the red color, it belongs to the beginning part. Okay, so based on this, you are able to tell the relative position, and then you can further confirm by looking at the sequence over here. You know that the lettering here should be the same as lettering over here because they originally uh, uh, come from the same strand like this. So these are the overlapping part to confirm. So that concept is used for the uh, when you have different fragment but you are able to reconnect based on the similarity at the overlapping position. So that is the important part. The overlapping is the one that is letting you to confirm. Okay, and then of course the marker is also important. That will further strengthen your decision that these belong because they are overlapping, because the marker also show they should also be there based on the information you already have. Okay, so that is how it is done in terms of sequencing. But problem arise when some of the YAG inserts contain DNA from more than one chromosomal location. This has complicated efforts at generating a YAG contig map of human chromosome, because sometimes, you know, for example, in this example, you can have a marker that is quite unique, which means that all of the marker are different. But sometimes you can have a marker that can be similar at 2 or 3 position. So when that happen, then you are unable to determine, okay, this one, this fragment, and okay, this, for example, like I have another chromosome, uh, three different three different chromosomes, but all of them have the same marker. Okay, all of them have the same marker. Okay, but then I don't know whether it belongs to the position, the same position or not, because they have the same marker. Although these are, in this case, theoretically, if they belong to the same original position, I will be able to arrange them to become, for example, like this, because the same marker, right? This one will be like this. Okay, so because they are the same marker. But then the sequence over here are different because actually this marker uh, can be located at 3 different positions on the chromosome. For example, on the chromosome, the same marker sometimes located, okay, on the same chromosome at 3 different positions. So I don't know which one belongs to which. I need information from other fragment that is overlapping with them in order to confirm the position. Okay, so that is problem arise when some of the inserts, yet insert this clone contain belong to the more than one position. Okay, so many labs have switched to the bacterial artificial chromosome vectors with capacity of 300 kilobase and the ability to replicate in E. coli as a resource for their sequencing project. So you remember the one that I have drawn just now, this one, one contact map, one contact map. So this one, all of them belong to different clones. Okay. So what happened is that with the Y chromosome, this one, the length here is very long, and then it is very difficult to get the entire length. So it is problematic, so complication. So what happened is that you can cut into smaller pieces and then clone into, you call it bacterial artificial chromosome. But the concept is still the same, whereby you have the fragment clone into the bacteria chromosome. Okay, and then you have a shorter version of the original contact map. So you can do sequencing and then you assemble based on the overlapping again. Okay, overlapping again. All right, so that is how it is done. So when a high resolution map is available, sequencing is possible. So basically just now I jumped the uh, gun a little bit because this is mentioning about the sequencing. So what happened is once you have the fragment overlapping, you clone, then you can do sequencing and the overlapping will let you know the relative position. So this one is considered high resolution map. So because you always have very detailed marker position, and when you have this, so what happened is that this marker is a known position, right? So all of these are known. So when you cut into smaller pieces, this the the thin line, all of these are the unknown. So when you have the entire sequence, you can start to reassemble by looking at the overlapping and also by looking at relative position of the marker. Okay, so now after you have the map, then you are in a clone, you will do the sequencing of each individual sequence nucleotide. So that is previously you have a dideoxy sequencing method. Okay, so dideoxy sequencing is a method where you do a sequencing. So on the DNA strand. Okay, now we go into more detail. So this one is actually from the clone just now. I don't want to use the red color. I want to use the blue one. Again, back to blue color. Okay. So so when so this one is a clone, isn't it? The contact the yak or back clone. Back or yak. So this one is the DNA on it. So when you want to do sequencing, so you will do you want to determine the letter, the nucleotide, like the ATGC sequence of this strand. Okay, so double helix, but you only need to determine only on one side because from one side you are able to determine the other side, the complementary strand, right? So to do a sequencing of this, you do a dideoxy sequencing method. Okay, so dideoxy sequencing is used to to do the DNA sequencing. Oh dear, sorry. So DNA is synthesized. DNA is synthesized from a template and terminates with the incorporation of fluorescently labeled dideoxy NTP. Dideoxy NTP. Here. Okay, you recall the nucleotide structure. Can anyone recall what is in the nucleotide in terms of the chemical constituent? What?
Pentose sugar,
Yes.
Phosphate group,
Okay.
nucleotide
nitrogenous
base.
base.
All right. Well done. Well done. So you have a phosphate group, and then you have ribose, not pentose, yeah, ribose sugar, and then you have nitrogenous base, right? So this one is basically a nucleotide. So you have a nitrogenous base, and then you have a phosphate group over here. So you can see over here at this one is carbon position number 1, carbon position number 2, carbon position number 3. Um, you have uh, OH over here. This one is a normal nucleotide. This one is H over here. Masih ingat tak struktur ni kan? Okay, so based on this, so this will be when you want to do DNA strand, then you continue, let's say, the other downwards, then the phosphate will, you remove the hydrogen, the phosphate will do phosphodiester bonding, then this one will have the another nucleotide, isn't it? And then this one will be H, this one will be OH. And then the continuation of phosphodiester bonding can happen because of the OH is over here. But if let's say I remove the oxygen over here, uh, just this nucleotide contain only H, so it means that this one is you don't have the O just now. The normal nucleotide don't have O, but this abnormal nucleotide, I remove the O. It also do not have O. So when this happen, you have I remove no oxygen from over here, no oxygen from over here. So you have mono and di. So di means two, isn't it? So dideoxy, meaning to say that I don't have the oxygen molecule at this position. So when I don't have the oxygen molecule, this phosphate nucleotide cannot come and do reaction in order to form the phosphodiester bonding. Okay, so that is why in the sequencing method, in addition to the normal nucleotide that is floating around, which you call it the normal nucleotide, you call it DNTP. Okay, DNTP, which consists of what? DATP. What else? DTTP, DGTP, and DCTP. So ATGC, the deoxyadenine triphosphate, adenine, thymine, guanine, cytosine, all of them, when they are together in one tube, we call it the DNTP. Deoxynucleotide triphosphate, so DNTP. Okay, so another molecule we call it dideoxy NTP because remember at the position here they don't have the oxygen. They don't have the oxygen, so dideoxy. So when let's say in a nucleotide, when this one is 3 prime, 5 prime, if the growing DNA is growing, when if I add in dideoxy, then the fragment will stop over here. Okay, and then another round reaction. When I do the reaction, if this one all the normal nucleotide comes in, but somehow dideoxy comes in. at this position. Then I have two strand that is different in one size nucleotide only. Okay, for example, this one is, let's say, 6 nucleotide, this one is 7. So one nucleotide difference. So eventually, based on this, you can have it grow until long. So then you have a series of sequence whereby the dideoxy stop over here. Dideoxy, dideoxy. So that is the concept of sequencing using the dideoxy sequencing, or in another word, you call it uh, Sanger sequencing. Okay, so please, if you are not that clear of what I am trying to explain, you go back afterwards. What you can do is that you can search for Sanger sequencing. Sanger sequencing, or probably you can type dideoxy sequencing in the YouTube. But the concept is that it grows by one and then it stops. So eventually you have collection of increasing size, and based on that, if you okay, just now, let's say this is the DNA template, and then if I grow it, it goes increasing of size. The difference is only on one nucleotide. Okay, this one one after another. So this one will be this, and then this, and then this, because it cannot go further because the polymerase cannot add in because of the what, dideoxy, isn't it? Okay, so increasing by one nucleotide resolution. So if this one is the nucleotide deoxy that is added here is DDA, for example. This one is DDG. This one DDC. So when I do an electrophoresis in a tube, so this one will go in first. Oh dear, what happened? Uh, if I do electrophoresis, okay, I do electrophoresis. So the smallest one will go in first. So what happened? This one will go in, and then this A actually have a blue fluorescent. My laser detect here, so it goes here, and then it detect A, and then this one goes in. This one is G. G carries the green fluorescent, for example. Then the laser detect G. Then it says, oh, G. And then this one go in. What letter is this just now? Let's say this one is T just now. So this one it detect T, and then this one it detect T again. So this is how the process. So now you know that the sequence over here actually is AGTT. So each one goes in. Actually, the difference is by one size resolution. So now you can have the letter. So this is how the nucleotide is sequenced. So one sequencing reaction limited to about here until 500 using the Sanger sequencing method. Okay. Any question? Please ask question. I know you are quite blur a little bit. Tanyalah soalan. Tak faham. While I try to erase all this. Soalan please. Oh, saya pula minta kamu tanya soalan. Okay. Okay, good. Now got plenty of question. Because we are able to tell the position of the fragment and rearrange them, can doctor explain what is the purpose of doing the sequencing? Like, why we do this? Is it like for determining possible mutation in the chromosome, for example, many position? Because, okay, the question, basically how I understand the question is that we are able to tell the position, right? The relative position of this. So based on the relative position, then you remember I mentioned just now the 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 squared one is the one that we know the information. The thin line we don't know, so that is the sequence that we want to know. So with the sequence that we want to know later on, because we know the marker, but we don't know. Okay, let's say you know that, okay, this region actually is for cystic fibrosis. Okay, initially you only know about the marker. You only know about the marker, right? You only know about the marker, but you don't know about the sequence here. So now, if I ask myself, oh, we know that this gene, the area from here until here, covered by let's say 1, 2, 3, 4 marker, actually is the cystic fibrosis gene. What actually is the sequence of the cystic fibrosis gene, so that I know what actually is the problem that is causing cystic fibrosis. Then I do the sequencing. That's why we do sequencing, so that we know the entire sequence. Then based on the sequence that we know, then we do analysis. Oh, this sequence actually, um, if we translate it, it is translated into a structural protein transport that is transporting the ion from one position to another. So now you know cystic fibrosis is because of the defective the ion transport protein. So that's why you want to do the sequencing. Okay, that is for example. Of course, yeah. So it is like to determine the possible mutation. All right. Okay. Irfan, you need some time to digest everything. Okay, take your time. Okay, but don't take it too long. All right. So basically, so that is why we do sequencing. So you remember in previous lecture, uh, let's say scientists already know this region cause when there is a defective region, it cause disorder kan. So we want to find out further. Uh, what else or where else is the disorder? You can see hundreds of different lists of disorders based on single mutation. Um, Based on single mutation. So based on that, we want to know the sequence so that we can make medication based on the information that we have, for example. So that is why originally scientists want to study the human genome or the human genome sequence, right? So and so that is basically the original answer, because in the United States they were doing the study on the individual uh, genes, for example. But then the question is, okay, now we know several genes causing disorder. So what else? What are the other genes that causing the other disorder? So because of that, then let's say, why don't we sequence the entire human genome so that we can find accurately what is actually the sequence of human genome. So if we detect the gene, we will be able to create a medicine from it. So that is why people start to devise a method to do sequencing and so on. Okay, so you just imagine like this one, 500 reaction only can be done. So you have to do many different sequencing reaction in order to get the entire length. So progress of the human genome and other project has been accelerated by improved technology for sequencing and analysis. Okay, for example, last time it is using the dideoxy or Sanger method. It is quite slow. So they develop technology whereby instead of doing one reaction can determine 500, they increase to one reaction can do 1,000, and then one reaction can do 10,000. Eventually, one reaction can do millions nowadays. Okay, so that is how progress has been ongoing quite some times already. So improve technology, improve sequencing. So human genome sequencing by mapping approach use bacterial artificial chromosome, but back insert is far too large to sequence in one reaction. So instead, the inserts were each sequenced using a shotgun approach. Okay, so you have to remember, if you have time to read even in the Wikipedia about the history of human genome sequencing, what happened is that after I mentioned scientists decided to study human genome sequencing, they sequence using the bacterial artificial chromosome or using the back or sometimes the bigger one using the yak. So back and yak. But uh, when you want to cut them into smaller pieces like this, you want to clone each of them into different vector, different plasmid. It takes quite quite sometimes. It is quite slow, basically. You remember this is only one chromosome. We have 24 chromosomes, and then each chromosome you have to partially digest. This one is, if you can count, I didn't count, is more than 20. But this one is just example, but actually hundreds of clones, thousands of clones that you have to do individually before you start doing the sequencing. So you just multiply so slow, okay? So even during my time, we are still using the dideoxy sequencing. So it's quite slow. So you just imagine. And then at that time, uh, the technology of doing it faster is quite expensive. So progress, improved technology it is improving, but it is quite expensive. Okay, so what happened is that they they want to make it faster. So this one, for example, is quite long. This one from here until here is quite long. So what happened is that they instead of they do the sequencing using the back and yak, they cut it into smaller pieces and then do each fragment individually without the need to clone the pieces into uh, without the need to clone it into smaller vector. Okay, so when you cut into smaller pieces, okay, for example, they started using this, and then another group comes in. Not only that, they start to cut this into smaller pieces, they start to cut the entire chromosome into smaller pieces, right? So that is the second group who did the sequencing. So you have to remember, in human genome sequencing, the mapping approach is the one doing it methodically, whereby they cut into quite not so small, and then they clone into a vector of back and yak, and then start doing sequencing. And when this one is too large, they take the vector, cut into smaller pieces, and doing the sequencing using shotgun. Eventually, some group came and started not doing this anymore. So they didn't do this. They just started to cut these smaller pieces straight away. All right. So and the one that they cut straight into smaller pieces, that one we call it shotgun sequencing. Okay. Because with a shotgun, you remember, with a shotgun, when you shoot, it's scattered everywhere. But then you have to then reposition back the chromosome. You just take something you throw away, but then you have to piece it back together, which one belong to which position. So that is how the shotgun process is done. Okay. So, Doctor, how does scientists actually found all those markers? Are they all doing those trial and error? More or less, the concept is yes. Because the question asks how, before we start to do the sequencing, the we start to do the sequencing, the question is how do scientists determine the markers? Okay, the markers have to remember this one is happen over a long period of time. Okay, it's not like when scientists decide to do a marker. Okay, let's do human genome sequencing. Okay, then only we start to do a determination of this individual squared marker position. Actually, before that, you have to recall when Watson and Crick start to determine the DNA structure, and then Masterson and Stoll, every McCroyd, McCarthy, eventually they determine the gene, right? So like work by Thomas and Morgan determine, okay, if there is a problem with eye color, it is located chromosome 1. Wing color, wing shape, located on chromosome number 3. So those information is being used by all the scientists later on. So some of the markers has already been known even before they know the DNA sequencing, but they know if we use this marker, if we put the color to the marker, if it hybridize, then we know that it has this information related to the gene. Okay, so the marker belong to this position on the chromosome. So experiments actually determine the marker. Many ways to determine the markers, but so how do scientists found all those markers? Just like the one I mentioned over here lah. By fluorescent in situ, restriction digestion. So those are ways to determine the marker. Okay, okay. Coming back to so to the shotgun sequencing. So each insert is cut from the vector, sheared into fragment that will be partially overlapping and cloned into a plasmid vector. Okay, so with the human genome sequencing, they start to have a better idea. So the large clone, for example, like this one, they start to cut into smaller pieces, and then they cut into smaller pieces. So they know that, for example, if you take this one, actually it's not one strand only, right? So these are all many strands, hundreds and thousands of strands of the same strand. So partial, when they cut into smaller pieces, partial digestion can also happen, in which some of them are overlapping, for example. Okay, so that happen, they clone into plasmid. So plasmid can play around with a smaller size so that they can do sequencing better. So sequence overlap are used by, so you can see over here, they start to use computer to determine the sequence of the DNA from the long one, we cut into smaller pieces, and then they are actually overlap. Some of them are overlapping. Okay, so based on that, they are able to determine the relative position so that the entire sequence now can be determined. Determined. Okay. So in theory, sequencing context for total length of 6.5 to 8 times the genome will span more than 99.8% of the genome sequence. So basically, is that um, if the length of the chromosome is 1 million base pair, right? So if I sequence it, my sequencing result produce 8 million base pair, meaning to say 1 million is the the physical distance, physical size, 8 million base pair. So I have to do sequencing of all these pieces representing the entire chromosome 8 times the length. So in order to cover 99.8% of the entire genomic sequence of the chromosome. Okay, so 6.5 to 8 times the length in order to make sure that all the overlapping are covered. Okay, all the overlapping are covered with the multiplication of the sequencing. So in practice, the Human Genome Project did its sequencing 7 times over. So 7 times over, and has obtained 97% of the genome. So of the chromosome 1 until chromosome 22 XY plus XY, it covers 97.7%, 97% back in 2003. Almost the same time, more or less the same time I complete my PhD the year earlier. Okay, so you can see that it takes, if we study the genome size, we can study the genome, estimate the genome size, 3 billion base pair. So the actual number of sequencing they did was 3 times 7, 21 billion base pair to make sure that all overlapping are covered. Okay, so that is how it is done. All right, the next way to do the sequencing is by a direct shotgun approach. So direct shotgun is the alternative to the the mapping approach. So we have one is mapping approach, and then number two way of sequencing is by direct shotgun. Okay, so you have to remember the mapping approach is a oh dear, uh, in a very specific methods. Lidah tergeliat already. But then direct shotgun, you do uh, sequencing, you cut into smaller pieces, and then you sequence randomly. Then the question will come to you, how then you want to arrange? Because uh, we have to determine, okay, if this one become fragment number one, number two, number three, and so on. So how do you know that this sequence, this fragment belongs here and not here? This fragment belong here and not here. Let's say, for example, now I got hundreds of fragments. How do I know that this belong to here, or is it belong to here, or this one, is it here or is it here, right? Because the way they do the sequencing is very short. Okay, so not even cover the entire marker region, for example. Okay, so mapping approach is a unique way in which they will do sequencing of the fragment and then reassemble using computer algorithm. So computing technology uh, was used when they do the overlapping sequence to do the overlapping sequence analysis. Okay, so the shotgun approach obtains genomic sequence by breaking the genome into overlapping fragments for cloning and sequencing, and then using the computer to reassemble it. Um, Advances have been made. This approach because of better computer algorithm for assembling the sequence and also the automation of the actual sequencing. So when human genome start with the mapping approach, so this one is a mapping approach like this. When human genome start, you have to clone everything. So very tedious, and then got the assembly. Then you have to analyze based on the marker and then put the sequence to do it more or less initially manually and then with the computer. But with the shotgun approach, they cut into very small pieces. Okay, a pioneer of this approach is Craig Venter, the Celera Genomics, sequence fivefold the human genome to 97, similar like the human genome. So, uh, human genome project, shotgun shotgun approach versus sorry. Uh, Shotgun approach is the Craig Venter, and the other one is using the mapping approach. Okay, shotgun versus mapping. So they do also fivefold, and then there were some gaps which will determine later on. So direct shotgun sequencing involve mechanical shearing. So what is mechanical shearing? Hmm? What is shearing?
Shopping.
Cutting, correct. Yeah, just like you have you know, the shear at home to cut the chicken. Okay, a stronger chicken, right? So shearing, mechanical shearing. So what happened with the direct shotgun sequencing? They cut the DNA, but they didn't use restriction enzyme. They just put the DNA. Let's say I have a tube of chromosome one fragments, chromosome one, intact chromosome one. I just put on the sonicator. So sonicator will vibrate, and then because of very high intensity vibration, you will cut the DNA into smaller pieces. Okay, and then with the smaller pieces, I just take the size that is only certain size only. For example, like this one. they take DNA that is around 1.6 to 2 kilobase only. So because when you cut the DNA into smaller pieces, it can be still have a DNA that is quite large inside, 5 kb, 10 kb, probably 100 base pair. So very small or very large, but they can uh, can like do electrophoresis to fish out or to just get only about 2 kb size. Okay, so that is the one. And even that, you can see if this one is 2 kb, they only do 500 on this side, 500 on this side, the middle 1000 they didn't do. Okay, but then you have to imagine, because they are cut into smaller pieces, there should be another fragment that cover from here until here is 500 over here, 500 over here. So this 500 sometimes overlap, let's say 200 with the other fragment, but the other 300 over here is the new information which was not sequenced in this fragment, but now sequenced in this fragment. And then there's another fragment again, for probably, that is overlapping, let's say 100 over here, which overlap with this one. Then this one, the unknown, is determined because they are 500, this one. So you can see we can walk like that to determine. And then you have to remember this side already also got information. So this will more or less confirm whatever that is later sequenced by another fragment over here. So you can see, the overlapping. So these are done meticulously using the computer algorithm. So computer programming basically. All right. So it will do the overlapping and determine the subsequent sequence of the DNA. So uh, so the subsequent sequence in the center of the clone DNA are obtained from overlapping clone rather than directly. So you remember just now, it will be determined after the overlapping is obtained. So computer analysis give the sequence of most of the genome with gaps caused by sequence missing from the uh, library. So sometimes you can have overlapping, uh, let's say 100 kb, but then there were some region that were overlapping, so it becomes some unknown region over here. So this one can use the shotgun approach, for example. Okay, and then those 2 kb is further confirmed by second library of 10 kb, meaning to say that from here, oh, dah padam ke tadi? From the shearing just now by vibration, what happened is that in addition to they take the 2 kb, they also take the 10 kb one, so that same fragment but with the 10 kb they should return more or less similar sequence. Okay, so that is how it is done. So advances in automatic sequencing and computer algorithm allow the whole genome approach to be used with even large genomes. So at that time, these are the approach that was done, and so so they come to the point of accuracy of less than one error for every 1000 bases. So very low error that were done. So these are more or less how it is done. You take the DNA, you cut into smaller pieces, and then they can uh, screen out the unwanted sizes. They only take about 2000 kb, and then they can do the sequencing by uh, the clone is just to produce more copies of the same fragment, and that those fragments of the same copies are then sequenced only on both ends, and then those sequence are then aligned using the computer programming, so that the sequence will be determined. Okay, so that is how it is done. I think 4 o'clock already. I am going to stop here while I address the some of the question, okay, that was asked. Okay, but I'm going to stop sharing for a short while, and I'm going to share the QR attendance. Okay, the question that I see someone ask, what uh, how does scientists actually found all those markers? Okay, they already addressed that question. What happened to the